Hello! This is Chapter 6, Section 3, Structures of Organelles. I am Mr. Williams. You should already know this. But just in case you're wondering, this guy that's about to come in here is not me. This is Jacob?
No, James.
James! Golly, I always get it wrong. I always say his name's Jacob. I don't know why. Alright, anyways, let's move on. So structures and organelles. This is where eukaryotic cells that contain, stop staring at me, contain organelles that allow the specialization and the separation of functions within the cell. So this is actually a rather long PowerPoint. I'm going to try to zip through it as fast as I can just for the sake of time. I'm not going to get Buddy in there. And now I have Jared Bossy staring at me awkwardly while I'm trying to do this. So, essential questions. We have, what are the structures of a typical eukaryotic cell? And what are their functions? And then we have, what are the similarities and differences between plant and animal cells? This is your vocabulary. Again, make sure you get that done by the time I have assigned. Make sure you understand these. There's a lot of things we got to go through. If you've got any questions, this is the time to ask, Holly. This is the time to ask. If you don't know what a mitochondrion is, you need to ask. All right, so getting into the actual subject matter eventually when this thing finally loads. Ooh, cool. Oh, there it is. All right, cytoplasm and cytoskeleton. The environment enclosed by the plasma membrane is a semi-fluid material called cytoplasm. Think of it kind of like the goo inside, okay? It's what everything floats in. It's like a water solution. It is not it is a water solution that has a bunch of different things mixed within it. In a prokaryote, all of their chemical processes take place here. All of them, because remember, there's no membrane-bound organelles for them to be contained within. So they're all going to happen within that cytoplasm. Versus eukaryotes, the processes take place within the organelles of the cytoplasm, like what we see right here. Let's zoom in on it. So what we're looking at, when we get into it, we're going to start looking at this is our actual plasma membrane. And this is what we're going to be looking at when we look at these different things. So within that cytoplasm is where this is happening, but outside we have our plasma membrane taking care of everything. This is the basic structure of a eukaryotic cell. We got our microtubule. I'm sorry, that's stupid. That's not a eukaryotic cell. That could be anything. It is a eukaryotic cell. I'm being confusing. Sorry. Let's move on. So the cytoskeleton is supported by a network of long, thin protein fibers that form a framework for a cell and provide an anchor for the organelles. So this cytoskeleton is a very important thing. We have microtubules and microfilaments. Microtubule is a long, hollow protein cylinder that form a rigid skeleton for the cell and assist in moving substances within the cell. Microfilaments are thin protein threads that help give the cell shape and enable the parts of this or the entire cell to move. So there's two different types of functions, but they're very, very similar at the same time. So one gives shape, the other one provides a skeleton, okay? So that's kind of just, you got to grasp your mind around it. We'll try to look at it. You'll see some pretty cool stuff when we watch the videos on it. So as we get into this right here is all the different organelles that we're going to look at. So let's just zoom in. We're not going to do the animation. We have a microtubules. We have a nucleus. Realize this whole thing, this whole purple thing right here, that's our nucleus. Then we have our nucleolus, which is that right there inside. Cytoplasm, again, that's like the goo that everything's in. And a mitochondrion, which is the powerhouse. Vacuole, which is going to contain our water. Vesicles, they transport everything around. Golgi apparatus packages and delivers things across the cell. Ribosomes do multitudes of different functions. Plasma membrane makes up the entire cell. Your smooth endoplasmic reticulum helps build your carbohydrates and lipids for structural purposes or storage purposes at sometimes. You have your rough endoplasmic reticulum, which makes your proteins. It actually does translation, not transcription. And then you have your centrioles, which help in cell division. And then your lysosomes help dissolve things. So that's just a brief analysis of all the different things we have. Here's the nucleus. Nucleus, more specific definition, it contains the DNA. And that DNA actually stores, stop laughing at me, Derek. That DNA stores information in the form of DNA, of nucleic acids. That information helps make proteins through going through other ribosomes. It's surrounded by a double membrane. So we have our phospholipid bilayer. Yes, that's It says phospholipid bilayer, which means 2 layers which are connecting to each other. But it is not a double membrane. It is a single membrane with 2 layers in it. This is a double membrane, so it's got a lot more. We call that our actual nuclear envelope. Do you want to say hi real quick before you...
How can you even say...
Right.
Oh, there it is.
Yeah, it's right there. It's
I
right
just
there.
made this one.
All right.
Oh, it's backwards.
No, ribosomes are organelles that manufacture proteins, not membrane-bound organelles like everything else. These are actually just big proteins inside the cell, not necessarily huge, but they're a protein, and they do multiple different things. And you can find them inside the nucleus, or they're produced inside the nucleus. They help me produce other things in that nucleolus. So, that said, Angel, get out of the way. Why are you in the way, man? You act like I let you do that or something. All right, so getting into the next section, we have those ribosomes, and we also have the, just turn it in, we have the endoplasmic reticulum. Endoplasmic reticulum is a membrane system that of folded sacs and interconnected channels that serves as a site for protein and lipid synthesis. Again, 2 types. Rough synthesizes your proteins, smooth synthesizes your lipids. It's called rough because it has actual ribosomes embedded in the membrane. Versus the other one, the smooth, does not have the ribosomes blended into the membrane. So we're going to skip that one too. The next structure is the Golgi apparatus. It's a flattened stack of membranes that modifies and sorts and packages proteins. These proteins that are packaged into sacs, we call that a vesicle, and it can fuse to the plasma membrane and release proteins to the environment. And actually remove waste the same way. When you package something into that vesicle, vesicles are like the post office, man. They transport everything wherever it needs to go. Once this loads, we'll continue, but I'm waiting for it to load. All right, next one is vacuoles. Vacuoles are a membrane-bound sac that's used for temporary storage. They store food, enzymes, and waste. Vacuoles are only found in plant cells. That is not correct. Sometimes you can find them in animal cells because they're everywhere. But animal cells don't necessarily need them as much. Vacuoles use that to increase what we call turgor pressure, which makes a plant nice and rigid and sticking out, versus no turgor pressure causes them to wither. So make sure you understand that. Lysosomes, they're vesicles that contain digestive enzymes. They help digest waste. They help, or not waste, they help digest excess or worn out organelles and food particles. They also are somewhat the immunity system of an actual cell. They help digest that bacteria and break down viruses that invade the cells. We'll watch something as we get into viruses called Battlefield Cell that you will really enjoy. I know it. And then we have our centrioles. Centrioles are made up of microtubules that function during cell division, and they are located in the cytoplasm. It's actually been discovered if you remove the centrioles out of the equation, the cell will still divide. So it's still kind of, we think they help in cell division, but they're not necessarily required. And then we have the mitochondria. Mitochondria is huge. I was talking about it in the last video about endosymbiotic theory on how the 2 prokaryotes, one ate the other and used the other one started helping it out, so it didn't kill it. And this is what that is the mitochondria or mitochondrion. It takes fuel particles, which is food, and turns it into usable energy, into ATP. They have an outer membrane and a highly folded inner membrane that provides surface area to help for breaking the bonds of sugar molecules. Sugar molecules are the food particle. Then we have chloroplasts. Chloroplasts are in plants and in some other eukaryotes, and they capture light. They convert it into a chemical energy called photosynthesis. So that chemical energy, that's actually the formation of glucose, of some of those carbohydrates. It takes CO2, 6 CO2 plus 6 H2O, input sunlight. It's going to convert that into C6H12O6, which is sugar, and 6O2. Yeah. So it takes all that, and we try to put it all together, and it's really, really interesting. So they use multiple small discs called thylakoids, which those contain the actual light-capturing pigment. Then we have a cell wall. Plants and fungi are things that have cell walls. Animals do not have cell walls. Plants have a cell wall made out of a carbohydrate called cellulose. It is a thick, rigid mesh of fibers that surround the outside of the plasma membrane. It is not the plasma membrane. If I have a cell wall, I still have a plasma membrane. All things have plasma membranes. And in this case, it is made up of a carbohydrate called cellulose with plants. And then we have cilia and flagella. Cilia are short, numerous projections that look like hair. They move in tandem like oars on a rowboat. That's where we have all these things around. Those are cilia. Versus flagella. Flagella are longer and less numerous than cilia. They move in a whip-like fashion. They're both made up of actually microtubules, and they help propel the cell and help push things into the cell as well. The stapler, there's no staples. We're not going to watch that. We'll do that in class. Just put a paper clip on it. Comparing cells, we have plant cells have things that animal cells don't, such as chloroplasts and chlorophyll and vacuoles and cell walls. And then we have, all right, so we have the part with the organelles on what's different between plant and animal. Next thing we got is how these organelles work. The understanding of different organelles allows for us an understanding of cellular processes. This is protein synthesis, for instance. It begins in the nucleus with the information contained in the DNA. Then we convert that DNA into RNA. The RNA leaves and ribosomes leave the nucleus and produce protein in the rough ER, the endoplasmic reticulum. Then they go from there. Those proteins that are produced in there, they're sent to the Golgi body to be packaged and sent where they need to go. Those packaged proteins are then delivered to other organelles where they serve a variety of functions. So that right there should be the end. Awesome it is. All right, cool. I just did 24 slides in 11 minutes and 55 seconds. It's legit. Get this one done. I'll try to get the next video done for you soon, and you guys can watch that and have it finished. Hasta luego. Tengo un buen día.